Welcome to Dr. Glynis' Chicken Genetics series, where today we'll be considering the gene for fibromelanosis. Fibromelanosis causes an abnormal accumulation of melanin, giving black skin, eyes, muscles, and internal organs. Even the bones appear to be black due to them being covered with a membrane which is pigmented. However, although I've seen it suggested on the internet, hens with fibromelanosis don't lay black eggs. Two of the best-known fibromelanotic chicken breeds are Ayam Cemani and the Silkie. If you associate the stunning Ayam Cemani chickens with fibromelanosis, then you can be forgiven for thinking that fibromelanotic chicken breeds also always have black feathers. However, this isn't necessarily so, and you can see this clearly if you realise that white Silkies can also be fibromelanotic. They show hyperpigmentation of the comb, wattle, face and beak, and also have intense pigmentation of skin and internal tissues. Fibromelanosis most probably originated in Chinese chickens and occurs commonly in some parts of Asia, especially Southeast Asia, where they are important for folk medicine and play a part in their religious beliefs. Old Chinese texts from the time of the Tang dynasty, somewhere in the region of 12 to 1400 years ago, describe birds with black skin and fluffy silky type feathering. Around 730 years ago, Marco Polo, who lived for some years in China, also gives enthusiastic accounts of such chickens. The fibromelanosis locus is on chromosome 20 and therefore autosomal. The fibromelanosis allele involves a complex chromosomal rearrangement, including two large duplications and an inversion. These result in the upregulated expression of the endothelin 3 gene that is responsible for melanin hyperpigmentation. In Ayam Cemani chickens, the gene is considered almost completely dominant, so it is very hard or impossible to tell if a bird is homozygous or heterozygous just by looking at it, although sometimes heterozygous birds may have a reddish-black leakage in the comb. I've noticed a similar thing in Silkies sometimes, but I was told by a DNA testing lab that all purebred Silkies are homozygous for fibromelanosis. I did wonder if eye colour was a clue to heterozygosity, perhaps with homozygous birds having black eyes and heterozygous birds having dark brown eyes. However, there are other possible explanations for this, including interactions with other genes. If you have any thoughts or observations about this, please feel free to leave a comment below. The expression of fibromelanosis is known to be influenced by modifying genes, especially the ID gene. The recessive wild-type allele is needed in the homozygous form to allow dermal melanin to show, leading to the blue-black skin and shanks in fibromelanotic birds. This complex locus is located on the Z chromosome, with recent research pointing to the methylthioadenosine phosphorylase gene as a strong candidate for its function, but possibly in conjunction with another gene or genes. Being on the Z chromosome, it might be that female birds are affected more than male birds with darker legs and skin. And if you have any thoughts on that matter, please comment below the video. There are also other genes that affect fibromelanosis, possibly including the W gene that determines skin colour. The C locus, whose recessive allele causes recessive white, and the extension locus, which affects the base colour and pattern of chicken plumage. I did see a paper that indicates the black fibromelanotic birds of Chinese origin may have a unique extension locus allele. Some fibromelanotic breeds are known for their local adaptations, including increased fetal viability and an innate immunity against microbial and viral infections. One interesting paper on an Indian fibromelanotic breed called the Kaduknath suggested that the fibromelanotic gene is closely linked to a bactericidal gene. This illustrates genetic linkage, where alleles of genes located close to one another on a chromosome are typically inherited together, and it is something we'll come back to in another video. In this case, the close physical linkage of the two loci means that this breed of chicken, which has been selected for fibromelanosis, has the added advantage of being resistant to various pathogenic illnesses. The allele for the bactericidal gene increased in frequency not due to natural selection itself, but because it is linked to another gene that is undergoing a rapid increase in frequency due here to artificial selection by humans. The bactericidal gene is said to be hitchhiking with the fibromelanosis gene. This is an important phenomenon because it is not just advantageous alleles that can do this. Sometimes strong selection, which is typical of artificial selection, can inadvertently lead to an increase in a linked gene with deleterious effects. The deleterious allele gets dragged along and increases in frequency because it's physically linked to the gene that's being selected for. This phenomenon is known as genetic draft and shows how selection on one gene impacts neighbouring genes, potentially causing harmful alleles to spread and persist. In summary, the fibromelanotic gene is a dominant autosomal gene whose expression is influenced by other genes, including one that is sex-linked. It causes the abnormal accumulation of eumelanin in various parts of the body, but not in the feathers. It is especially common in birds of Chinese and Southeast Asian origins, and some breeds may have increased immunity to infection due to an allele that hitchhikes with fibromelanosis. Thank you very much for watching this video. If you have found it useful, please like, comment below, and share it with your friends or on social media. Subscribe so that you're notified of chicken genetics videos as they come out. I hope to see you very soon.